At the beginning, I would like to thank you, the organizing committee, to invite me. This is a long way for me to come. And thank them for organizing this symposium or conference on the ultrasonic vocalization. Many years ago, it was just, this line of research was created as a curiosity research, but these days it is one of the most important lines of research in behavioral neuroscience, because it is about mechanisms of emotions and explaining how emotions work. I would like to present, to show in my presentation, this importance of discussing the emission of 22 kilohertz vocalizations, which are emitting, showing the negative emotional state and are equivalent to human crying. So I wanted to raise a couple of questions. First of all, briefly review how those calls are emitted and what are the brain mechanisms or brain systems which are causing this dramatic response. And then answer briefly at the end what is the biological reasons that they emit them. So let's begin from the beginning. The calls are emitted. I will be pointing with my arrow. I hope you see that. So when the rat is emitting 22 kilohertz calls, it assumes a specific posture. It lowers the head and aligns the head with the trachea and with the rest of the body. Here on the right-hand side is the normal posture of the rat, so the head is a little bit lower than the trachea. Here is trachea, here is larynx with vocal folds, and here is the buccal cavity. So it aligns that, then stays motionless, opens the mouth. Most of the sound is coming through the mouth, minimal component through the nose. So the buccal cavity is the channel for emit them, and builds up the pressure in the abdomen. You see here the bulging abdomen pushing the air through narrow opening in the larynx. It's only about 1 to 2 millimeters opening, and it squeezes air through that opening with very, very long vocalizations. About the length of those calls, I will be talking in a moment. The first question was how the rat knows where is the about 22 kilohertz. He doesn't. What he does, he's lowering the emission of 22 kilohertz calls to the lowest possible level. So at the beginning, we see tuning, and you see that here, 2 emitted calls. And the first one, here is the enlarged, this first call. You see the gradual tuning to that desired frequency. And desired frequency is the minimum frequency the rat can emit in ultrasounds. The first call is tuned. The next one is already, he's already in the appropriate level. We have proven that each call was divided into maximal set of frequencies and minimal set of frequencies. And if we correlated intercall intervals, here is the intercall interval, with the maximum frequencies, it was a very highly significant relationship. That means the longer is the distance from the previous call, the rat starts with higher frequencies. And when he continues, then the frequency is lower. But when we correlated intercall intervals with minimum frequency, there's no relationship, because the minimum frequency is constant, always the same. In literature, you find very often that it is 18 kilohertz, 19, 20, 21. I suspect it is not less than 20. In this case, 658 calls were not lower even than 21 kilohertz calls. The biggest interest is on the length of those calls, why they are so long, and some other are very short. Here are examples of some short calls from even less than 50 milliseconds, 200, 300, and we call them short calls. But all of them are within the same frequency band. There are some intermediate, like 470 in this particular case. This is not short and not long. And then there are some very long. The longest we shown here is 1 second. Yeah, 1.1 second. I think it is 1 digit too much here. So the question is how we divide those calls and whether we can divide them into short and call. This is from practical point of view. The short calls can be from 0.1 to 0.3 seconds, that means 100 to 300 milliseconds, and we suggested that 300 milliseconds is the dividing level, and long calls are from 500 milliseconds up to the longest we ever recorded in the lab, 3.3 seconds. You do not realize how long it is. I will just show you that in a moment. This is crazily long. Some rats emit shorter calls. For example, most wild rats caught in the wild and recorded in the lab will be about 600 milliseconds. In the lab, depending on the technique, depending on the environment conditions, the length of the call changes. The sonogram or the spectrogram of that crazy long call is here at the bottom. This is, I suspect it is the maximum the rat can do. If we look at the lung capacity, mouse has about 1 milliliter, rat has 10 milliliter lung capacity. I'm not saying that this is the all air used for vocalization, but general capacity of lungs. Humans has about 6 liters. To demonstrate for you how long is that call, I can use linear logic and say if the 10 milliliters of air created 3.3 second call, then if we could shrink our vocal folds to 1 millimeter opening and use our 6 liters, 1 hour call would be about half an hour. Don't try that at home because we won't have audience for tomorrow. Do people have such calls? Yes, they do. Infant crying is like that. They cry with extremely long cries up to the complete loss of oxygen. They are driving themselves into hypoxemia, and even skin on the face is bluish from lack of oxygen. Usually around the mouth of small crying children, you can see so-called circumoral cyanosis. So this is not only rats. We all, when we cry, we may use those calls with such an enormous length, basically emit as long call as possible. Why is it happening like that? To answer that question, I would like to show you the brain system which is driving those calls, which is forcing the rat to emit such tremendously long calls. Everyone is very well familiar with the ascending reticular activating system, which is shown here in yellow, which is innervating the entire mantle, entire cortical mantle, but it's not much known about the other specific ascending mesolimbic systems for emotional arousal. There are 2 parallel systems, one shown in green, which is cholinergic, one shown in reddish, which is dopaminergic. Dopaminergic system drives 50 kilohertz calls, and the cholinergic, the 22 kilohertz calls. So we suggested that there are 2 parallel ascending mesolimbic systems. One is cholinergic, shown here in red. One is mesolimbic dopaminergic system, shown in blue. It doesn't mean that those are all cholinergic neurons in the brainstem or all dopaminergic. This is only a subset of them, which is used for that arousal, emotional arousal. But this is very specific subset of cells, and I would like to talk about them for a couple of minutes. Since the main topic of this talk is the emission of 22 kilohertz calls, I would say that they originate from laterodorsal tegmental nucleus, more specifically cholinergic neurons of laterodorsal tegmental nucleus. Those are very specific neurons which are communicating between themselves within the nucleus by gap junctions and creating electrical coupling. So if one begins to fire, all other begin to fire. And the whole system is activated very powerfully. This is in more detail than that ascending mesolimbic cholinergic system originating from laterodorsal tegmental nucleus. It has two leaves. One leaf is dorsal, mostly innervating thalamic areas. The other leaf is ventral, going down to the most limbic systems, most limbic structures, hypothalamus, up to lateral septum, and even areas known in the past as renencephalon, and narrow path is going to the prefrontal cortex. This system is driving cholinergic, is driving emission of 22 kilohertz calls, and it is entirely cholinergic. Acetylcholine is released here literally everywhere. I will just explain why everywhere. The system was not noticed and not much discussed in literature because we couldn't see it. Those fibers, cholinergic fibers, are so thin and so thin and divided that it is difficult to visualize them. In electron microscope, of course, it was possible to see them, but no one has seen entire system. You will not believe, but it has taken about 70 years to see that system. The first paper showing the entire system appeared this year for the first time. This is how it looks like. It is by Zhao and co-workers. This is only a couple of neurons on the left-hand side, and you see that their projections are weird. The projections are going everywhere. All what you learned in neuroscience is not applied here. You, of course, learned that there is neuron, a couple of dendrites, one axon with synapses at the end. It is not like that here. From the main cell body, the axon begin to branch. One axon can branch up to 1,000 times, and the branches are still branching further. So this is a bush. Actually, the best term is the bush, cholinergic bush going. This is only a couple of neurons, and you see that they are creating that bush, and narrow path going to the prefrontal cortex. On the right-hand side, it shows different neurons because they are not have those trees identical. That bush is, for me, very amazing. It reminds me the witch's broom. What it is, in botany, this is the outgrowth on the trees, which is shown here in the insert. When from the trunk, there is not one branch going, but thousands of small branches. All are alive, but creating such a broom, witch's broom. And this is really a witch's broom in the brain. And look, it's not going everywhere. It is in the subcortical limbic structures mostly. Here is a fragment from electron microscope. It's even difficult to see how complex it is. This is innervation of cortical area, showing it looks like a mold with tiny and branching and branching. Now, to make story more complex, there is not only final terminal synapses, synapses on the whole length of those axons, on those entire bush of axons. They are varicosities. Each varicosity can create synapse, and each varicosity releases acetylcholine. So here is the example of such axonal varicosity on cholinergic axon, and black arrows indicate some of them which are active, and the arrowhead shows the terminal. So there are terminals as well. Also in the insert, there is a lot of varicosities, and one is terminal. So the characteristic of that system is that they not only create such bushes, but the synapses are everywhere. So if the one neuron is active, it releases acetylcholine in an enormous amount, literally in the all subcortical limbic areas. And to make story even more complex, this is not classical neurotransmission. There are three types of transmission here. One synaptic, which is shown here. The varicosity creates typical synapse and releases acetylcholine into postsynaptic area. But there is also some spillover. There is more acetylcholine release, which can go to receptors on other axons, not creating synapses with varicosity, in this case with GABA neurons. And there is classical non-synaptic release, which is release of acetylcholine from varicosities, which can travel through the cerebrospinal fluid to whenever are the cholinergic receptors. So if all of that is firing, all of that is released, what is happening? The limbic brain is overwhelmed by that release. And this is the essence of creating emotional state. That's why emotional state is stopping all activities, and we really look funny, and rats too, when that emotional arousal system kicks in. So we tried to map that crazy bushes of axons, and we're injecting the carbachol, which works like acetylcholine. And it appeared that we can induce 20 kilohertz calls from entire that system. Whenever we inject it, there is vocalization. The large dots indicates more calls, smaller dots, less calls. This is from all that system which I showed you at the beginning. Up to the lateral septum, here are the black dots showing the lateral septum from which we could induce calls. Now the question is what is the mechanism of that? A long time ago, pharmacologists told me that I am weird because you cannot have the same pharmacological response from half of the brain. Well, I said, yes, I did not invent that. This is how it is. And it is specific. It is dose dependent. It could be to carbachol, and it could be reversed by atropine very significantly and very strongly. Those are muscarinic receptors responsible for that first response. Here we have again recreated that bush. And look, this is only subcortical areas, the exactly area which we mapped with carbachol. So question again, how is it possible injecting carbachol to one spot and have response from the entire bush? Here for simplicity, I just showed one line for the ascending mesolimbic cholinergic system. And what is happening, the laterodorsal tegmental nucleus, when it is active, it acts as a whole by electrical coupling. So what happens when the laterodorsal tegmental nucleus is active? The whole system is buzzing. It's really overwhelming our limbic system. Well, it's clear, so we know how the emotional arousal, negative arousal appears. But what about injecting carbachol? If we inject carbachol into one spot, carbachol is working only on one area, local area. So why the whole system is active? And it appeared that it works in such a way that whenever we inject carbachol, it's connected by loops with the laterodorsal tegmental nucleus. Loops back and forth, and one loop is enough to activate entire laterodorsal tegmentum, and entire system is buzzing. What we know about those loops? We know about those loops quite a lot. It was suggested almost 70 years ago, but neuroscientists are strange people. They sometimes neglect some results. It was the Wally Nauta who suggested that and proven that by neuroanatomy. He was neuroanatomist. By the way, I pronounced his name as he pronounced his own name, Nauta, but all English-speaking people pronounce his name Nauta, which is incorrect, but we know what we talk about, whom we talk about. This is his diagram showing the limbic midbrain area within this laterodorsal tegmental nucleus and ventral tegmental area are located, and all other limbic areas, including frontal cortex, were called forebrain limbic system. And he said, here are the loops going back and forth, labeled with thick arrow, because this is very heavy connection. So he, in 1958, told us how that system works and how, on what basis it is activating the limbic system. So this is how the emotional states are created. It's not only the self-propelling system of lateral dorsal tegmental nucleus by electrical coupling, not only by massive innervation and branching and varicosities and massive release of acetylcholine, but also those loops continuing that system for a long time. We ask next question. If we inject different doses of carbachol, we may have different intensity of that response. Here are diagrams, histograms, showing length of individual calls collected from large number of couple of thousand of individual calls. Here you see for 0.25 microgram, half, 1, 2, and 4 micrograms. If we inject it in the same rats, it works, but if you inject into naive rat right away 2 or 4 micrograms, that is epileptogenic threshold level. But if gradually is done, they do not get seizures. And we notice that there are 2 groups of peaks, we call P1 and P2, very short calls and longer calls. Some of them are extremely long, here up to 2 milliseconds in this case. 2 seconds, sorry, up to 2 seconds. So assumption was if the higher is the dose of carbachol, the stronger is the response. And we expected that the stronger is the response, the longer will be the call. No, it is not like that. Here is on the left-hand side, correlation of the duration of calls. Dose of carbachol, and it is decreasing. That means the stronger activation by carbachol, the shorter the call. Okay, so why they call with long calls? At the same time, bandwidth with the doses is not changing because they have to emit always the same characteristic call. Here, I analyzed the very long calls, more than 1,000 milliseconds and less than 1,000 milliseconds. And with the increasing doses, number of longer calls is clearly decreasing. And the number of calls then shorter than 1,000, number of calls is increasing. So they emit more shorter calls when the system is activated by higher dose of carbachol and less longer calls. Now the sound frequency is not changing with the dose, and rightly because they are emitting specific call, which has to be specific acoustic parameters, and it keeps those parameters. They are kept within the increasing doses, but the intensity of the calls is increasing with the dose of carbachol. How then we can explain that that sometimes are so long calls and sometimes are short calls, if this is not an intensity of the emotional arousal? We came to the conclusion, but this is not tested. I'm leaving that for you to test. This is the situation, circumstances, presence of our other rats, and the nature of threat. They emit 22 kilohertz calls as a threatening stimulus. But if this is done, it's not really life-threatening situation. Rats will tend to emit very long calls. For example, the famous 22 kilohertz post-ejaculatory vocalizations, which created a lot of misunderstanding in literature. Everyone thought that this is maybe a sign of pleasure. No, it is not. The male is emitting those calls after successive copulation, and this is signal to the female, leave me. It cannot continue. an experienced female after those 22 kilohertz vocalization leave. Only unexperienced continue soliciting sex, and this is not what the male wishes. In this case, those calls, when the female is not obedient, the calls sent by male are extremely long. Well, it's not life-threatening situation, but I would say it's annoying. Other situation is air puff. The rat knows what we are doing. The rats are intelligent enough that this is experimenter, which is puffing them with the air in the nape of behind the head, and they hate it. And we continue and continue, and the rats begin to call, probably to us, with extremely long calls. But in other situations, like I said, wild rats, when they expect more danger, they emit shorter calls. In rats addicted to cocaine, they emit, if they don't have the drug, they emit massively 22 kilohertz calls, but they're short, because that comes mostly from the internal need. There's no external danger. I don't know. I'm just giving you a couple of situations which possibly could explain when the rat emits shorter calls and longer calls. The shortest calls induced by us were where we injected glutamate directly to lateral-dorsal tegmental nucleus. This is the strongest stimulation you can imagine. 99.9 neurons is depolarized by glutamate. So all cholinergic neurons were strongly depolarized in lateral-dorsal tegmental nucleus. That was the strongest activation of arousal system. And believe it or not, rats were calling with enormous number of those calls, but short. They were rushing, short after, as fast as they could. So it is not intensity, it is something else. I don't know how to call it. It has to be experimentally shown. And the final question of my talk is why they call, why they cry like that, because those are equivalents of crying. And the same question will be for human infants. Why they cry so intensively and so longly? We suggested that this is a specific system which evolved very early in evolution, before the dopaminergic system for reward evolved. So this is older time, and evolved from the interaction between mother and infant. And during that evolution, so that happened probably more than 12, 14, 15 million years ago. So the rats specifically perceive those vocalizations, and they have capability to induce automatically aversive system in the recipient. So if the child is crying, the mother, as soon as she hears the call, is in the aversive state. That automated system of inducing aversive system, activating that arousing cholinergic system, we call etotransmission. You can find the details in the current opinion of neurobiology 2013, and there is a full definition of etotransmitter. It is by analogy to neurotransmitter, not any substance which can depolarize neuron. This neurotransmitter, similarly here, not any acoustic dangerous sound, is the etotransmitter, only the specific calls. I will not take more of my time because I was asked to leave some time for discussion. So I just wanted to summarize that aversive emotional state is initiated by ascending mesolimbic arousal system releasing acetylcholine. I would say massively releasing acetylcholine. Those are diffuse ascending cholinergic projection with numerous varicosities, really numerous. 1 cubic millimeter has millions of those varicosities. And release acetylcholine in a large limbic region and diencephalon in forebrain. The mesolimbic cholinergic system has self-propagating properties by loops between forebrain and midbrain, and aversive arousal caused by 22 kilohertz vocalization in rats is equivalent to human crying. Duration of individual calls is highly variable and may convey the intensity of the negative emotional state with the along with the number of emitted calls. And ultrasonic vocalization are capable of inducing emotional state in the receivers, and this is really fast. In humans, it was calculated that we need 500 milliseconds to realize what stimulus is coming to us. We hear it, but to realize what it is takes about 4-5 milliseconds. Ultrasonic call or baby cry is already recorded in the cortex within 50 milliseconds. So this is automated fast response, which almost like a pre-programmed system is inducing emotional state in the receivers. So this is very interesting system and very, very old from evolutionary. I would like to acknowledge some of my collaborators. I listed only those from whom I've taken the results and the support from different foundations. Thank you for your attention.
Great content. So there's a real interest in this group on looking at social behavior of animals in a large group, in large stage interactive. And I want to remind us all of the work of Robert Blanton and his work with 20-tailed vocalization, and sort of your thoughts on that of when a couple of things. When in a large cage, how those animals vocalize, and also how they can transition from making the aversive vocalizations to flight behavior. What are your thoughts?
First, about the calling in a social group. Blanchard has shown that social group is necessary to emit those calls. It doesn't mean that when rat is completely alone will be not emitting calls. It can, and sometimes they emit, but overall, when they are alone and they know there's no other companions, they are silent or emitting only a few calls. That audience is needed. So within the burrows, Blanchard's burrows, of course, they know that other rats are there, so they are very eager to vocalize. By the way, I can mention that in burrows, the ultrasonic calls are traveling further and faster. So this is not the call which is designed to communicate in the open spaces. So they do emit. Well, I don't know more details about which situation will call more calls or longer calls, but the whole system the Blanchard studied is helping to communicate vocally. Now that breaking points, which we are talking when the calls go into the flight, it's a big mystery known from a long time ago. There are even mathematical models using so-called catastrophe theory, which shows that the line going as a function to a certain point breaks at a certain point. What is deciding that break and what is making that break, we don't know. And I cannot answer you what it could be, which part of the cortex or the limbic system decides about that. It could be regarded as a form of panic, which can kick in at a certain point. And it's in humans, panic can kick in at a certain point or not. So I don't know more on that issue.
It's not for this great talk that really connected a lot of dots for multiple decades. And I was wondering, I mean, I guess you probably would have shown it, but do you know whether there is any study that directly show particularly the disabilities in the level of those momentum by probable injection or another form of activation of the living system?
Yes, there are. We published a lot of papers from activating that system. Usually it was by glutamate because it's more effective. But there are cholinergic receptors, and it could be induced by carbachol from lateral dorsal tegmental nucleus as well. those branching neurons, which I mentioned, creating the bushes, start right away after the cell bodies. So already within the lateral tegmental nucleus are branches and connections.
Thanks for a very thought-provoking detail. This is Paul Clark. I think we should probably introduce ourselves for the Zoom exhibit. I'm thinking that the brain in a mouse is built on the same principles, or measurement system, etc. So what do you think would happen if we injected carbachol in mice, given that they don't have the
Well, I tried to convince a lot of people working with mice to do that experiment, but no one was brave enough to try. What I would suggest from other studies, which when aversive states were induced in mice and ultrasonic calls were recorded, that the overall frequency of calls follows the rule in rats. Higher frequencies are associated with positive state, lower frequencies with negative state. So what I would expect carbachol would do will induce the lower frequencies. It has to do something. I'm not saying that absolutely every mammalian species will vocalize after injecting carbachol. I agree that there are some species which do not have this kind of response because we do not really know how the transposition from that cholinergic arousal works to initiate call itself. But as distant species as cats have exactly that response. So I would suggest that rats that mice will also have. People are probably discouraged, as you said, they don't have specific 22 kilohertz calls, but the overall shift in frequency is in that direction. It will be an experiment.
We have a couple of questions from Zoom, and just to you are explaining the system with really minimal abilities, so sorry for the inconvenience, especially this very sorry. But right now we got two questions from Zoom, one from Stefan M, and hope I pronounced well. And the question is about strain differences. What rats are you using, or were you using, and do you think there are strain differences which are relevant?
Yes, there are differences. There are individual differences, quite a big differences. With Jeff, we tried to separate those tendencies in rats, and you can have rats which heavily vocalize and rats which are not very eager to vocalize. There are differences between strain, not whopping differences, not extremely strong differences. 22 kilohertz calls is always 22 in all strains, but there are some differences in level of frequency, tiny, in length of calls. So we cannot deny that there are not differences, but the main type of those calls remains the same.
Okay, and we got the feedback. Thanks for your answer. And second question is about the vocalization that contains both components like 22 and 50. Did you solve it, and what is the possible meaning of those?
Yes, it can happen like that in many situations. This is not common, but it can happen. This is to give you a brief answer. This is the same as we see in children who are crying for some reason, and then we try to explain them that there is not really a reason to cry, and they begin to do something strange. They laugh and cry at the same time, briefly. This is like the system it does not know what to do, and rats can do that. The most clear situation where I've seen that is when we inject carbachol. Carbachol works for a long time. It has about 100 times stronger and longer action than acetylcholine itself. So after injection of carbachol, the response is very long. rat is calling and calling and calling and calling. At certain point, what the brain is doing is applying brake to that. That means changing the aversive state into more positive state. And the rat can interject the 50 kilohertz calls between the 22 kilohertz calls for a brief moment and then stops calling. So those are mixed situations. This is not, I'm not saying that this emotional state, that we can have emotional state with both systems working together, mixed state. I don't think this is the case. What happens, those systems are working very fast. So what the brain is doing is in aversive state, then switches to positive and switches to aversive, switches to positive, switches to aversive, and eventually has to make its mind which system has to dominate. But those are brief moments. Usually the animals and we also know in what state we are. It's good or bad. And then only one type of calls is needed.